Chromosomes and Disorders. We're going to begin at looking at some chromosomes. A karyotype is a display of chromosomes of an organism. They are identifiable by their size, so the chromosomes that would be from mom and dad match up by size. Centromere location, that's where the microtubules and such would attach to during chromosomal division. And then the banding pattern that we can see, the fluorescence of the banding pattern. Your chromosomes have 22 paired autosomes. Those are mostly your body chromosomes. And then there's 2 paired sex chromosomes. Those are not homologous, so there is not a similar derivation of those chromosomes. This is a karyotype. You can take a look and you can see that the chromosomes that are matched up do in fact have similar centromere location. They do have similar banding patterns, and they also are about the same length. Looking down at the X and the Y, those are your sex chromosomes, and you can see that they are not homologous like the remainder of the chromosomes are. Now, we can look at X-linked inheritance, and as the name implies, this is when a trait is carried on the X chromosome. Males usually exhibit the phenotype because the only chromosome that they receive is the X chromosome. Males have an X chromosome and a Y chromosome. So assuming that mom is going to give an X chromosome to the son, it has a good chance of carrying that gene. Examples of X-linked inheritance would be hemophilia. X with a dominant H is normal. X with a recessive or mutant H would be the phenotype that was expressed. Hemophilia, remember, is a bleeding disease. If you look at the second one there, we've got the genotype, X big H, X big H. And then we also have X big H, X little h. That's a female who is a carrier because they have the mutant gene, they just aren't expressing it. And then if a male ends up with X big H, Y, they're normal. However, in the event that the male ends up with an X mutant H or recessive H and a Y chromosome, they would be a hemophiliac. As well, a female could in fact become a hemophiliac as well. Her genotype then would be X little h, X little h. And that's just a little bit rarer of a condition to be female and be hemophiliac. Taking a look here, you can just see how this is passed along through this pedigree chart. In generation 1, we have a male who's normal and a female who's a carrier. So in the event that they have children, there will be one female who is a carrier in that group. There will be one male who ends up being a hemophiliac. Showing the female then who reproduces again, we could end up with more and more hemophiliacs down the line. This was considered to be the royal disease because they had lots of inbreeding, and as a result they kept passing this trait along further and further down the line. There are also some sex-influenced traits. These are not necessarily attached to an X chromosome, as in the X-linked traits, but they affect one sex more than the other. So baldness is an example of a sex-influenced trait. B positive would be normal, and B would be baldness. So a woman must have a little b, and have other factors to be bald. So baldness is not completely a male problem. Men are severely bald if they have the recessive little b, and have pattern baldness if they have a little b and a B positive. Testosterone also stimulates the expression of baldness, which I suppose is unfortunate because testosterone is also responsible for secondary sex characteristics in a man. And so you wouldn't necessarily want to suppress testosterone release. All right, and then we're going to look at the chromosomal number abnormalities. Trisomy 21 is known as Down's syndrome. The risk of having Down syndrome or having a child with Down syndrome increases with age. If you are under the age of 30 having a child, the chances of having a child with trisomy 21 is 1 in 1,300. If you're 40, that changes to 1 in 100. And if you're 45 or over, that changes to 1 in 25. So as you age, your chances significantly increase, which is why women who are above the age of 34, I believe it is, are considered advanced maternal age for having children. And this is just a picture of a young man who has Down syndrome. What happens with trisomy 21 is that the 21st chromosome ends up having 3 copies because we have what we call non-disjunction. So if you look at the top, on the left with A, we end up with the chromosomes separating, but they don't separate as they should normally, as on the right. What happens in that first division is that the chromosomes don't separate. So the gametes, instead of having 1 copy of a gene, end up having 2, which is where the trisomy comes from. So there would be a total of 3 when that egg or sperm came together with the other gamete. This could happen as well during meiosis 2, which is what's happening on the right-hand side. In meiosis 2, same thing happens. We don't get separation of the chromosomes into 2 daughter cells, and as a result, you have an extra chromosome inside of one of the cells. The other flip side to this is that the cells that don't have any genetic information can, in fact, fertilize an egg, or the egg can be fertilized as well. And so it could be that you have extra chromosomes, or it could be that you have missing chromosomes when non-disjunction takes place. All right, alterations in the number of sex chromosomes. One Y chromosome will be the male phenotype. So as long as an individual gets a Y chromosome, they will appear physically to be a male. One situation is called XYY, or double Y syndrome. In this case, since there's Y chromosomes, the individual will be male. They're going to be tall, and they would have some impaired mental capacities. Klinefelter syndrome is XXY. Again, since this individual has a Y chromosome, they are going to be male. They're very tall, and they're sterile, so they're unable to reproduce. Additionally, individuals with Klinefelter syndrome do develop breasts because of that extra X chromosome. In some cases, they have those breasts removed. And then the other alteration is XO, or Turner syndrome. In this case, the O represents a cell that would have had a lack of chromosomes or no DNA. So this phenotypically is a female because there's only the 1 X chromosome. These individuals are short. They end up with small breasts. They additionally are sterile. And Turner syndrome ends up being fairly rare. So if you take a look at this, the individual on the left would have Klinefelter's syndrome, and the individual on the right would have Turner syndrome. We also have situations with abnormal chromosomes. There can be deletions where genes are actually deleted off of the chromosomes. The chromosome piece may break off and then be lost. Something called Cre-de-Chat is a deletion problem. These individuals are mentally and physically retarded, and Cre-de-Chat basically means cry of the cat, and that's because infants with Cre-de-Chat sound, the cry that they have sounds like a cat. Translocations ends up being another problem. This is when a chromosome piece breaks off and attaches to some other chromosome, or maybe the same chromosome, and translocations are found a lot in cancerous cells. And then there are some genetic disorders as well. PKU, phenylketonuria, is a homozygous recessive problem. These individuals lack an enzyme that's used to metabolize an amino acid called phenylalanine. 1 in 12,000 births end up with PKU. These individuals may become mentally retarded, and it's really as simple as diet. If the diet doesn't include any PKU or phenylalanine, these individuals will not become mentally retarded. So a strict diet can keep these individuals normal. You may have also heard of something called Tay-Sachs. Individuals with Tay-Sachs are missing a lysosomal enzyme to digest lipids. The lipids accumulate in the neurons and cause them to die. This is a really sad disease because the person will die by the age of 3 or 4, and up till about the age 2, they appear very normal. Everything's going fine. They're developing normal. They're hitting milestones, and then unfortunately at age 2, things start going downhill quite quickly. Huntington's disease. This disease begins in your 30s. It is a dominant lethal allele. So if a person has a copy of the dominant gene, they are going to unfortunately come to a demise as a result of Huntington's disease. It is a progressive nerve degeneration type of disease.